In this series of screencasts, I would like to introduce you to the physiology of the respiratory system. And in this first set of sessions, I would like to cover these learning outcomes. I will describe the components, organization, anatomical, ultra, and cellular structure and functions of the respiratory system. And I hope that after listening to this screencast and also after completing a online exercise supported by our new LT platform, you will be able to understand and define the lung volumes and capacity, and also start appreciating the use of diagnostic tools to measure lung function. Further information will be provided in due course about this exercise supported by LT platform. Which are the functions of the respiratory system? Well, first of all, to exchange oxygen and carbon dioxide between the environment and the cells of the body. What do I mean by saying that? The respiratory system is aiming to supply, to provide our cells with oxygen that is essential for cellular respiration, but also is aiming to dispose, to get rid of the carbon dioxide which is produced by the cells during the cellular respiration. Another function of the respiratory system is also to regulate blood pH. There are four processes composing the external respiration, which are summarized here. And here a cartoon to help you to visualize, if you like, the different processes of what is called external respiration. So the first process is the pulmonary ventilation. So the process that allows the exchange of gases, so oxygen and carbon dioxide, between the atmosphere and the lungs. The second process is involving or consisting of the exchange of gases, so oxygen and carbon dioxide, between the air that is in our alveoli and the blood that is present within the capillaries in the lungs. The third process of the external respiration consists of on the transportation of oxygen or carbon dioxide in the blood between lungs and the rest of the body, so the tissues and the different cells. The fourth process of the external respiration consists of the exchange of gases, so oxygen and CO2, between the blood and the cells within the tissue. As I mentioned earlier, the CO2 is a product of the internal respiration. This cartoon is summarizing the anatomical components of the respiratory system that consist of nasal cavity or oral cavity, larynx, which is the part above the trachea, which divides into main bronchi that will divide further within the lungs into bronchi. You have two lungs which are encased within two layers of tissue. The inner, so the visceral pleura, and the outer or parietal pleura. These are two membranes that secrete fluid into the pleural cavity surrounding each of the each lung. Then you have the diaphragm, which is included in here as an anatomical component of the respiratory system, as you will learn later on in this screencast, is important for the actual physics of respiration. But the diaphragm in the respiratory system is a dome-shaped sheet of muscles that separate the chest from the abdomen. I'd like to add these two pictures here to show you which is the morphology of our lungs. So if you take a look at the cast of human lung here on the left-hand side, you have a, a, a structure that can shows you the airways in the right lungs and the pulmonary arteries and vein in the left part or in the left lung here. In the picture on your right hand side, you have a scanning electron myograph of a silicon rubber cast of human pulmonary acids. And it's interesting to look, well, to look at this silicon rubber to understand which is the structure of the lungs or of the alveolar ducts. Here, indicated by this arrow, you can see the transitional bronchiole, and you can see also the first order of respiratory bronchioles here. And then you can see also that the alveoli are actually, are covering the alveolar ducts, which are underneath of the alveoli here, those little sacs. It's interesting how they create these rubber cast. In order to create this model, lungs need to be harvested from a human cadaver, and the lungs are actually injected with silicon. The silicon is then allowed to harden before the tissue or the lungs are macerated in potassium hydroxide and then treated with boiling water and hydrogen peroxide to then detach the tissue from the polymer. And what is left is this cast of the human lung that help us to understand the structure of bronchioles and alveolar ducts and alveoli. In this image here, an explanation, the picture actually of a trachea is a micrograph of the tracheal wall that shows you which is the composition of the trachea. So in the trachea you have a layer of mucosa composed by goblet cells, but also pseudo-stratified ciliated columnar epithelial cells. You have a lamina propria as part of the mucosa. You have a submucosa in here, which contain some glands. And then you have the cartilage underneath of the submucosa. Looking further on the histology of the respiratory system, the type of tissues present in each part of the respiratory system changes with the size actually of the different parts of the system. So you have a trachea which is composed by muscles, contains glands and cartilage, and the lumen range between 20 to 22 millimeter of diameter. Then you have bronchi which have still a certain amount of cartilage, but reduced amount of cartilage. There is a layer of smooth, a continuous layer of smooth muscles here, and you have still some glands. But then you reach the bronchioles, and as you can see, the cartilage is completely is not present, is not expressed anymore. There are no glands. There is only a non-continuous layer of smooth muscles. And also the diameter of bronchioles is very reduced when compared to trachea. Continuing on looking into the histological changes that occur in the respiratory system, I would like to point to note that the respiratory system can be actually divided in conducting zone and respiratory zone. We will talk about these two different zones in a second. But I would like you to note that the conducting zone is characterized by a very thick epithelium, and the size of the epithelium decreases across the respiratory system, and it becomes very thin in the respiratory part or zone of the respiratory system. In the conducting zone, you have goblet cells, ciliated cells, glands, cartilage, as I mentioned earlier. You have a thick layer of smooth muscles, and you have elastic fibers. But as you can see, when you reach the respiratory zone, you lose the majority of the cells that I mentioned earlier. And the respiratory zone, or the alveoli, or I should say the alveolar duct, the alveolar sac, and the alveoli, they contain a thin layer of epithelial cells, some smooth muscle cells, and elastic fibers only. I mentioned that the respiratory system can be divided in two zones, the conducting zone and the respiratory zone. They are different not just because of the structure, not just because of the type of cells they are expressing, but also because of the function that they are mediating in our respiratory system. So the conducting zone is responsible for bringing the air into and out of the respiratory zone. And the respiratory zone is essential for the exchanges of gases between the atmosphere and our lung. And the zone is the area that contains structures involved in oxygen and carbon dioxide exchanges. So starting with the conducting zone. The cells expressed on the conducting zone are the following. Ciliated cells. They are important. They are cells that present cilia. Cilia are elongated, motile, cylindric projection from the apical cell membrane. And they are important because they can remove inhaled particles and remove the mucus upward toward to the higher part of the respiratory system, so to the higher part of the trachea, so it's easier or it's possible to get rid of the mucus. There are also goblet cells or mucus secreting cells. They are important to secrete mucus, which is essentially in trapping particles. They are accidentally inhalated or present in the respiratory system, and once the mucus is trapping these particles, then the ciliated cells can help the excretion or the getting rid of the mucus. You also have smooth muscle cells. They have sympathetic and parasympathetic innervation that regulate the airways dilation and constriction, so bronchodilation and bronchoconstriction. We will talk about that in more details later on. But just for now, remember that smooth muscle cells have sympathetic, can receive sympathetic fiber, and via, thanks to the beta-2 adrenoceptor expressed in this smooth muscle, sympathetic innervation can mediate bronchodilation. Then you have parasympathetic, as I mentioned, innervation, and in these smooth muscle cells, and thanks to the presence of muscarinic receptor, the parasympathetic innervation can mediate constriction of the smooth muscle, so bronchoconstriction of the airways. So not only the type of cells present or in this conducting zone are typical of this conducting zone and allowing this area of the respiratory system to perform the different functions for which this conducting zone is responsible of, but also I would like to point your attention on the table here, where is summarized the presence of cilia, smooth muscle cells, and cartilage, or I should say, is shown how the presence of cilia, smooth muscle, and cartilage is actually changing across the conducting zone from trachea, bronchi, and bronchioles. Last but not least, another type of cells you can find in the conducting zone is the club or Clara cells, particularly expressed in the terminal bronchioles, so at this level. They are exocrine secretory cells. They secrete the primary component of extracellular substance lining the respiratory bronchioles. They also regulate the content of secretion. They also act as a progenitor cells for themselves, so for Clara cells, but also for ciliated cells. So they act as a stem cell. Another key role is in the biotransformation of harmful and toxic compounds introduced to the lung via inhaled air. So the club or Clara cells, they also have an important role in biotransforming or detoxifying and therefore protecting our airways from harmful and toxic compounds such as furan, hydrocarbons, ozone, other elements of tobacco smoke, for example, and other substances. What about the respiratory zone? The respiratory zone is characterized by a different type of cells, as we already discussed earlier. Respiratory zone expresses fibroblasts, which are elastic fibers, and epithelial cells, which are very typical of the respiratory zone. They are called type 1 and 2 pneumocytes. And a type 2, please do remember this one, they produce surfactant. We will talk about surfactant later on. But the surfactant is important to reduce the surface tension of alveoli. In the respiratory zone, you also find phagocytic cells, also referred to as alveolar macrophages. The function of these cells is to keep the alveoli free from dust and debris. They are phagocytotic cells. They keep the respiratory zone clean from these, as I mentioned, dust and debris. Again, I like to point your attention to the table here. And if you think about the cilia, smooth muscles, and cartilage, you can see that there are some cilia in the respiratory bronchioles, but cilia completely disappear from the alveolar ducts and certainly from the alveolar sac. There is, as I mentioned earlier, some smooth muscles in the respiratory bronchioles and alveolar duct, and there is no cartilage in this part or zone of the respiratory tract. Here a cartoon just to help you to visualize the type of cells that are present in the alveoli. We mentioned the type 2 alveolar cells. They are important in secreting the surfactant, which is found within the fluid lining the pulmonary lining the inside of the alveoli, here in light blue. And then you have alveolar macrophages, and the alveoli are embedded in a mesh of capillaries, as we will talk about in a second. And you can see in here pulmonary capillaries and erythrocyte located within the capillaries.